this video, we'll talk about something very interesting: synthetic biology. You remember Bob the Builder and how he uses amazing tools to create architectural marvels? Well, we all know what robots are. Robots are machines programmed by humans in such a way that it performs the task it is instructed to. Can we program living cells in a similar way? The answer: yes, we can. The programming of these living cells is called synthetic biology. Synthetic biology is a field of science that involves redesigning organisms for useful purposes by engineering them to have new abilities. Synthetic biology researchers and companies around the world are harnessing the power of nature to solve problems in medicine, manufacturing, and agriculture. Redesigning organisms so that they produce a substance such as medicine or fuel, or gain a new ability such as sensing something in the environment, are common goals of synthetic biology projects. Some examples of what scientists are producing with synthetic biology are microorganisms harnessed for bioremediation to clean pollutants from our water, soil, and air, rice modified to produce beta-carotene, a nutrient usually associated with carrots that prevents vitamin A deficiency, yeast engineered to produce rose oil as an eco-friendly and sustainable substitute for real roses that perfumers use to make luxury scents, among a plethora of other applications. Well, don't get confused between synthetic biology and genetic engineering. There is a thin line of distinction between the two. In synthetic biology, scientists typically stitch together long stretches of DNA and insert them into an organism's genome. Whereas, in genetic engineering, scientists typically use tools to make smaller changes to the organism's own DNA. Synthetic biology is definitely something that is going to take over the world really soon. It has the potential to solve many contemporary problems. Let's look at some commonly used terms in synthetic biology. Biobricks: standardized DNA sequences that can be used as modular building blocks to rapidly create more complex sequences. You can imagine these like Lego bricks, which can be assembled to create large structures like buildings, cars, etc. Gene expression: the process by which gene information recorded in DNA exerts its function. Recall the central dogma? Yes, proteins, the workhorses of cell. This is how they are produced. Recombinant DNA: DNA that contains sequences of DNA from sources that were brought together by techniques of molecular biology and not by traditional breeding methods. Vector: plasmid or modified virus into which a DNA fragment of interest is integrated, and this carries the DNA of interest into the host cell. Finally, computer-aided design: the use of computer software in the creation of design. In synthetic biology, it can be used to design DNA sequences. If you like this video, give it a thumbs up, subscribe to our channel, and stay tuned for more such content.